This is Chapter 6 Notes, Part 3, all about the third and final step of cellular respiration. I'll warn you, it'll be quite intense. To remind you of where we left off, the idea of cellular respiration is to break apart a glucose molecule to release the energy that's stored in the bonds so that the cell can make ATP. The cell will then use ATP to store ATP or to get proteins to change their shape and do some kind of job. The first step of cellular respiration involves glycolysis, where a glucose molecule is broken in half to produce two pyruvates. Glycolysis occurs in the cytoplasm. If the cell has both oxygen and a mitochondria available, those pyruvates will then enter the mitochondria to do step number 2, which is the Krebs cycle. The Krebs cycle involves more rearranging of the atoms within those pyruvates to release a little bit more energy. During glycolysis, a net gain of 2 ATP are made. During the Krebs cycle, also a net gain of 2 ATP are made. However, even more important than those ATP are the electron carriers that get made. During glycolysis, we make an electron carrier called NADH, and during the Krebs cycle, we make more NADH, but also a new electron carrier called FADH2. Electrons have energy, so by carrying an electron, those molecules are carrying energy, which will then be used in step number 3. Step number 3 is like the big kahuna. It's where you get the most bang for your buck and where the most ATP are produced. So this video will be all about that step number 3. Step number 3 is called oxidative phosphorylation. The phosphorylation part comes from the fact that we are going to phosphorylate ADP to make ATP. The oxidative part of the word comes from something that is going to lose electrons. Remember, Leo, lose electrons, oxidation. The molecules that are losing the electrons and thus giving this oxidative word are NADH and FADH2. So those electron carriers will lose their electrons, and that energy will ultimately be used to make ATP. This third step of cellular respiration is pretty intense. So we're going to go through part of it first on this slide, and then we'll take a look at the rest of it. So we have electron carriers called NADH and FADH2. These electron carriers were made in glycolysis and the Krebs cycle. They are going to go into the inner mitochondrial membrane. The inner mitochondrial membrane. The mitochondria looks somewhat like a bean shape, and within that organelle called the mitochondria, there is an inner membrane that's zigzaggy, like shown. It is in this inner membrane where step number 3 occurs. These electron carriers, NADH and FADH2, arrive at the inner mitochondrial membrane. When they are there, they are going to deposit an electron to something called the electron transport chain. The electron transport chain is generally depicted as what looks like a staircase. On each of the steps, there is a protein. So it's like an analogy of a staircase. It's not actually shaped like a staircase, but each step represents a particular protein. When NADH and FADH2 encounter the first protein, I think I should label those. The proteins are called cytochrome C, cytochrome oxidase C. When NADH and FADH2 arrive at that first protein, they deposit an electron, giving that protein the electron, giving that protein a little bit of energy. In turn, when the electron is removed from those molecules, we have NAD and FAD, which can then return to glycolysis and the Krebs cycle to pick up additional electrons. So those molecules will get reused. Also, there was a hydrogen that is missing here because we've got the hydrogens here. So another waste product is the hydrogen. Those will come into play a little bit later. So my electron carriers, NADH and FADH2, are my shuttle buses that drop an electron off at the first protein. This then continues like a game of hot potato, where the electron is passed from protein to protein, going down the electron transport chain. The reason that it's depicted as a staircase is because as the electron gets passed from molecule to molecule, a small amount of energy is released every time. Each of these proteins in the electron transport chain is going to gain an electron and then immediately lose that electron. So it gains an electron and then it immediately passes on that electron. So every protein in that chain gets reduced and then oxidized. So what we are showing here is that the electron is getting passed down a series of proteins. This game of hot potato continues protein to protein. However, at the end of the chain here, there is a very important molecule that has to grab that electron, and that molecule is oxygen. So if there is no oxygen present, if you imagine there's no oxygen present, it would cause a giant traffic jam up this chain. So oxygen is required for respiration because we need it to collect the electron at the end of the electron transport chain. When that oxygen molecule grabs that electron, it will combine with the hydrogen to produce water as waste. So the entire purpose of you inhaling is to grab an electron from the electron transport chain. Lack of oxygen, giant traffic jam, nothing will continue. This is the first piece of step number 3. So continuing on, oxidative phosphorylation. We still have not made any ATP. Now we're ready to put the final pieces together. Oxidative phosphorylation is occurring in the inner mitochondrial membrane. So this is a 2-layer membrane. I'm inside of the mitochondria. Step number 1, as we just went through, of oxidative phosphorylation is the electron transport chain. That used energy carriers called NADH and FADH2. It produced hydrogen as waste. That hydrogen will come into play to create water here in a few moments. And then it returned NAD and FAD back to glycolysis and the Krebs cycle to pick up more electrons and continue to break down sugars. As that electron gets passed down the electron transport chain here, a small amount of energy is released. We're going to add on now the remaining pieces. That energy that gets released from the falling electron is actually going to be used to pump these hydrogens across the membrane. Those hydrogens will become part of water. We're just not quite there yet. So we have a huge movement of hydrogen ions across the inner mitochondrial membrane. We call that active transport. The hydrogen ions are pumped all to one side of the membrane. We then end up with a super high concentration of hydrogens all on one side of the membrane. We call that a concentration gradient. Which means there's way more of something on one side of the membrane. So we have a large amount of hydrogen all on one side of the membrane. Those hydrogen ions are all positively charged. And due to a law of thermodynamics that says that particles will spread out, like if I spray perfume at the front of the room, eventually the perfume smell will permeate throughout the entire room because those perfume particles will spread out. Same idea with this concentration gradient. We have a huge accumulation of hydrogen ions on one side of the membrane, so they are going to diffuse and spread apart. The way that they do that is through a giant protein called ATP synthase. ATP synthase is a very unique protein because it has a tunnel through the center of it. So those hydrogen ions will diffuse or spread out by going through the channel of that ATP synthase. As they are flowing through the tunnel portion of ATP synthase, they are carrying with them those positive charges, right? So as that protein encounters new positive charges, that protein starts to change shape. And it actually causes the base of the protein here to spin in a circle. So it's a very unique protein. It's like a tunnel with a spinning piece at the bottom. It kind of reminds me of like at a fancy restaurant, sometimes they come around with like a big, huge pepper grater, and they can spin the bottom of the pepper grater to add pepper to the food. So it's like the idea is you're holding the tubular piece and only the base of it spins. ATP synthase is then ultimately going to make ATP. So as the H plus diffuses through, the bottom of the protein spins. That molecular motion, the spinning motion of the protein, provides energy to jam a phosphate on to ADP. So we have ADP and a phosphate. As that protein is spinning, it provides energy to put that phosphate on to make ATP. We also have those hydrogen ions that diffused through, so they were up here, they are going through the ATP synthase, they do come out the bottom. So ultimately then I have my hydrogen ions that then join with oxygen to make water. So this is a very intricate process with a lot of things happening simultaneously. Just to kind of recap everything here. During glycolysis and the Krebs cycle, we made electron carriers. Those electron carriers have to be made in advance of oxidative phosphorylation. The mitochondria cannot break apart glucose on its own. So it needs those two earlier processes to occur. Those two molecules, NADH and FADH2, are carrying electrons and carrying energy with them. They deposit those electrons at the electron transport chain, where proteins play a game of hot potato, passing an electron on to their neighbor. At the end of the electron transport chain is oxygen. Oxygen is required to be there in order for the proteins to continue passing on. If there was no oxygen, my electron carriers would not be able to deposit any electrons. When those electron carriers drop off an electron, they release hydrogen. The hydrogen originally came from the glucose molecule. Those electron carrier molecules will then return to glycolysis and the Krebs cycle to pick up new electrons. As electrons are passed on the electron transport chain, they release a little bit of energy. That energy is used to pump the hydrogen ions all to one side of the membrane. You also might see them just simply referred to as protons being pumped, but more correctly, they're hydrogen ions. When all of the hydrogen is accumulated to one side of the membrane, we call that a concentration gradient. Those hydrogen ions of similar charges do not want to all be crowded together on one side of the membrane. So they will spread out and diffuse through a protein called ATP synthase. As the hydrogens move through ATP synthase, they cause the base of the protein to spin. The motion of that spinning provides energy to plug a phosphate on to ADP. That then makes ATP. We can get about 36, 34, 38-ish ATP made predominantly through this process of oxidative phosphorylation. So this is the moneymaker that's making all of the ATP for the cell. Those hydrogen ions join with the oxygen that was at the end of the electron transport chain to produce water as a waste product.